physical fitness and still not have reproductive fitness, right? An organism can be sterile but physically very fit, but that does not count as fitness when we talk about evolution or we talk about biological evolution per se. Is it clear?
Yes.
Yeah. Okay. Continue.
So then we discussed panspermia theory. So life according to this theory, like life according to early Greek thinkers, was that this was transferred to Earth in the form of a spores from the outer space. So this theory is still valid. This is a still valid idea. So then we did.
It's not a valid theory; it's a valid idea. So we cannot validate the theory, but it's still it's a very talked about idea in the field, especially among astronomers and astrobiologists.
Yes.
It's difficult to disprove this theory, because to disprove this theory, you basically have to disprove that life cannot come to Earth from anywhere in the universe, because there is no life in the universe. But that's impossible to prove that, because we cannot scan the whole universe with surety to say that there is no life elsewhere. Okay. Yes. Continue.
So then we did theory of spontaneous generations. So for a long time, it was believed that life came out of dead, decaying, and rotting matter spontaneously. But later, Louis Pasteur, he challenged this and he dismissed the above theory through an experiment that demonstrated that life can only arise from pre-existing lives.
Yes, and what was that experiment?
So we did it till here only.
Okay, we didn't discuss the experiment about the flasks. He took flasks.
No, sir.
We did.
Okay.
I saw the recording, so I know we did it.
See, we did Arpit. Okay, so what was there in the recording, Fatima?
There were two flasks, and one of the he took something. I don't remember what he took in the flask, but then he took it and he pasteurized it, and then one of the flasks, he covered it with a cotton cotton, and then and the other flask was like open. So he noticed that like the one which was covered, there was no bacteria or anything growing, but then the one which was open, in that like there was growth.
Yes, so basically he took a piece of meat, like dead, because flesh, yes, because that's a decaying matter. So one flask A, so there this piece of organic matter. So what he basically did was, so this is the plug, okay, on flask A, and there was no plug, it was open to air, so flask B. And in both flask A and B, there was an organic matter, which is flesh, okay? Organic matter, which is flesh or meat. Now, if you leave a flesh or piece of flesh or meat to rot, you will see that basically we were talking about this, that small microorganisms or maggots starts growing out of decaying, dead and decaying or matter, right? That's where this theory of spontaneous generation came up by this observation. But what he did, he heat sterilized flask A. Heat sterilized and then plugged it. And flask B was also heat sterilized and then left open to air. Now, because of this leaving, there can be spores that can enter and reach this meat and start developing, because there is nutrition, there is moisture, and if there is right temperature, things can start growing in this flask. But no growth will be seen in this flask because it is covered. So he proved that if theory of spontaneous generation was correct, then in both the flasks there are meat, pieces of flesh kept, and both flasks are kept under the same temperature and condition. It's just that one is open and the other is closed, but inside there is meat. So he proved that unless and until there is already some bacteria or spores existing on the flesh meat piece, there can not be life originating from that flesh. So life only originates from pre-existing life or spores of life. Is it clear, Arpit?
Yes, yes.
Okay, so this was the experiment. He basically so he did not use flesh, I think. He used yeast, dead yeast, because that's just easier to use. But again, it's a living organism which is dead, and so growth started coming out of it. We just, so do you want me to also dictate it, Harpit? Okay, write down.
Yes.
Yeah, write down. Louis Pasteur showed that in pre-sterilized flasks, flask which was plugged, there was no microbial growth. In the bracket you can write new life arising. There was no microbial growth, in the bracket, new life arising from dead yeast. Whereas in the other flask, which was open to air, life, new living organisms arose. From killed yeasts. New living organism arose from killed yeast. Is it clear? Written, everyone?
Yes.
Yes.
Okay. So basically, theory of spontaneous generation was dismissed once and forever. So it's not debated anymore because it's dismissed. Now, till now, we studied about certain theories that could or could not tell about certain aspects of evolution of life, but none of the theory actually give us any hint about origin of life, how basically life originated. If you believe it originated on earth, then how? And if you believe that it originated somewhere else, then how? Okay, so about origin of life, basically there were two scientists, Oparin and Haldane. And this is known as theory of abiogenesis. Very important theory with proofs, and the most accurate one till date. Now, the theory of abiogenesis was proposed by Oparin and Haldane. Okay, there were two scientists, Oparin and Haldane. Now, what they observed was, they didn't observe, basically what they proposed, they thought about life as the fundamental unit, and the fundamental unit of life, which is a single cell. So the earliest organism that could have evolved would have been a single cell, right? Makes sense, right? You cannot
Yes.
just assume all of a sudden a multicellular organism like human to come up. from nowhere, right? If life has to form or originate, the simplest one will come first. Yes or no? It's like anything that happens in nature. First simple things come up and then complexity start growing on it. Yes or no? So which is the simplest form of life present
Cell.
today? Yes, one
Cell.
single cell, right? A bacteria or something which is one single cell, small single cell. Now they said that life or living cells could have emerged from non-living things. Now, this was a very, very bold statement at that time, and also to many biologists and thinkers, it did not make any sense. How can life come up from non-living things? But if you just take it, read it as this statement that life arose from non-living things, that someone can, again go and argue that okay, this pen is non-living. Can it become living someday? That's not what theories mean when you read just the blank statements. So people who criticize scientific theories should read it and then criticize on the basis of the fact of it. But people who do not understand science and just read statements like human evolved from monkeys or life evolved from non-living substances, it's very easy to mock it and not believe in it. But you have to first understand the basic, what is the theory talking about in detail. So when Operin and Haldane said that life, so write down what they proposed, write down. Operin and Haldane proposed, Operin and Haldane proposed that life could have, now they were not sure of it, but they thought about it a lot. And they proposed that life could have evolved from pre-existing non-living organic molecules. And what are organic molecules? Name some organic molecules. Organic means that are present in living systems.
Proteins,
So
amino acids.
proteins, amino acids, these are organic molecules. But now question came, where did organic molecules came from, right? So right, and they also they also propose this to complete the theory. Life could have evolved from pre-existing non-living organic molecules, and the formation of life and the formation of life and the formation of life was preceded by what do we mean by preceded?
Something that
Preceded.
happened before.
Yes, succeeded means that comes after, preceded means that happened before. So the formation of life was preceded by chemical evolution. This is what the complete statement from theory is. Okay, and to understand what is chemical evolution, write down. Chemical evolution is formation of diverse complex organic molecules, formation of diverse complex organic molecules from inorganic components or from inorganic elements. Okay, or inorganic molecules, you can say. So you understood that they proposed two things. First, they said that everything that was required to make the first kind of simplest life form, everything came from chemical evolution. And then when these organic molecules were present, then they assembled to make the first life form, which is a unicellular single-celled life form. Okay. Now the theory, everyone understands the statement?
So can you explain again chemical evolution?
Yeah, chemical evolution is what I just dictated. Formation of complex, diverse organic molecules from inorganic molecules. Okay. So you know what is inorganic molecule? You have three divisions in chemistry: organic, inorganic, and physical. That you all must have studied, right?
Yes, yes.
So what do you study in inorganic chemistry? Arpit. It's a cross question
So-
to you. If you understand that, then it will be easier. Yes, tell me.
So the metals parts, metallurgy.
Yes, right. You read about inorganic earth metals, right? That
Yes,
are
sir,
found
yes.
from the earth. Yeah. So those kind of elements or molecules are not present in human body. Like they do not make human body. They are present in trace amounts, but they do not make the bulk of the human body or a cell, right? Cells are made up of nucleic acids, proteins, carbohydrates, etc. You do not find proteins in rocks or in earth, right? You only find proteins in living systems. Yes or no?
Yes.
Do you find
Yes.
carbohydrates on rocks or in water or in soil?
No, sir.
No,
No, sir.
right? So whatever molecules you only find in life, living cells are called organic, and whatever molecules and compounds you do not find in living cells or are also present along with living cells in the nature, like in rocks and inanimate matter, is called inorganic. So they said that inorganic molecules gave rise to organic molecules, and then organic molecules assembled together to make the first cell. The theory seemed very, very straightforward enough. Everyone understands the theory?
Yes.
But just because they said it, you're not going to believe it, right? Howsoever
How-
logical it may sound.
Sir, how
Yes,
did
yes, Swati.
inorganic molecules give rise to organic molecules?
That is what we are going to study now. That is what I'm saying. They just said it and you're not going to believe it, right, Fatima?
Yes, sir.
You're going to ask how, but not just how, you're going to ask proof. Because if someone says, you ask how, and I says that, these bonds go and bind there and this energy gets transferred, and due to a lot of radicalization and enzyme kinetics, inorganic molecules become organic. So will you agree? Will you now say, oh, some very, very difficult terminological jargons, so I better agree? This is not how science works, right?
Yeah.
Just because it is difficult or explained in a more complicated way does not mean it may be true. Yes or no? So how do we know that something is true in science? We are
Like
not going.
Proofs.
What are the proofs? A proof again can be a statement, no? How do you prove that a circle is a circle in mathematics? Exactly. The difference between biology and mathematics is also one of this. So you often get these questions, right, in mathematics, that prove that this is a circle, or prove this is an equilateral
Yeah.
triangle or isosceles. You cannot write that it looks like a equilateral triangle, so it's an equilateral triangle. You write some statements to prove it. You do some angle calculations to prove it. Yes or no?
Yes, sir.
Similarly, when you ask for proofs in biology, you are asking for experimental proof. Show me that what you are saying can happen, or show me a system where it is happening, or show me a organic evidence which can suggest that what you are saying must have happened. That's why this is the result. So these kind of evidences are asked in science. So when they said, the next question was, what is the proof? How can you prove that chemical evolution has happened? Now, for the same reason, to prove chemical evolution, then came an American scientist. His name was Miller. I don't know his full name, but and I just remember the part Miller.
SL.
What? Excel? I don't think it's Excel.
SL.
SL. Again, it's not the full name, no? But if you are sure, then I'm just writing it. SL. Miller.
Yes, sir.
Yes. So he thought of an experiment that can prove or disprove what Operand and Helden has proposed. So basically what they proposed, and in this theory, there were some other components. They also took into consideration when Stanley Lloyd Miller, okay, SL is better than, you remember SL Miller. So
Yes.
he said that, okay, on what basis Operand and Helden are proposing this theory? So the basis was that on early Earth conditions led to this chemical evolution to happen. Now there are two questions. Is this chemical evolution still happening? Like somewhere? We don't know. Maybe or maybe not. But is it happening in every water body around us? No, because now the water is very, very stable and the temperature of water is optimum for life. Fish swim in the water, so water does not have much energy. Okay, it has dissolved oxygen, it has dissolved nutrients, it has fish, and the temperature stays less. It's cool, right? Urey was another scientist, Fatima. It was not Miller Urey. The full form was not Miller Urey. Okay, it's called Urey-Miller experiment sometimes. Urey was actually working with Miller in the laboratory, so it's also known as Urey-Miller experiment. The idea came from Urey, but Miller did the experiment. Okay, Fatima?
Yes, sir.
Yeah, so I'm just using the person who was the backbone behind the experiment and not Urey. You are correct. It's called Urey-Miller experiment sometimes. Urey. Okay. So the basis of this experiment was that prehistoric Earth conditions were different from the conditions that are today on Earth. So this whole theory was based on that chemical evolution could happen at that time, at that point of time, because of the specific unique Earth conditions. So that's why we are not having proofs of chemical evolution happening now in our ponds and rivers and oceans, because the water is cool and it's very, very suitable for life to evolve. So Urey and Miller said, okay, fine, fair enough. So let's recreate prehistoric Earth condition and then see whether what you are proposing can happen or not, right? Fair enough. So what were the early Earth conditions? What could have been the early Earth conditions? So data, simulations, and scientific studies, both geological, biological, and chemical studies have suggested what could be the early Earth conditions like. Now, early Earth conditions were, first thing was the atmosphere was reducing. What do we mean by reducing atmosphere?
The temperature was decreasing.
No, reducing has nothing to do with temperature. Temperature was decreasing. Temperature was just unimaginably high. It started decreasing when the Earth started solidifying. That's a different thing, but still it was beyond life could sustain, like we can sustain. But that's not reducing, an atmosphere is not related to temperature. Okay, what is the word reducing means in chemistry? Reducing agent, you all have read about this.
Yeah, the addition of oxygen or
No,
removal of,
that's
sorry, addition of
oxygenation.
hydrogen and removal of oxygen.
Yes, so reducing atmosphere means that in the atmosphere the oxygen was either not present, or if it was present, it was very, very less or negligible, or it was in the form of some compound. It was not free oxygen at all. Okay, and reducing, that's why it was reducing, because hydrogen was there in bulk and hydrogen was present in almost all the compounds. So that is reducing atmosphere. So you can write down reducing atmosphere is low on oxygen and high on hydrogen. Low on oxygen and high on hydrogen. And reducing atmosphere contains what all things make an atmosphere reducing. It contains methane, it contains ammonia primarily, and of course hydrogen as a gas. And it also contains water vapors, but there is oxygen, but it's bound to hydrogen. So these all make the atmosphere reducing. Second thing that we know about early atmosphere is that the temperature was high. Okay, Fatima?
Yes.
Everything was melting and boiling. Volcanoes were just normal things, you know. Volcanic eruptions were not rare phenomena. They were happening almost everywhere, wherever there were volcanoes, okay, in the ocean or outside. So temperature was very high. And as I said, volcanic eruptions. And when the first cloud formation started happening, there were thunderstorms. Okay, so these were the early Earth conditions. Now they thought of creating these conditions in lab. Now how can you do that? How can you create early Earth conditions on a laboratory scale? To prove. Now basically, do you understand what they had to prove? What they were trying to prove? They were trying to prove the chemical evolution part. Everyone understands that it is possible that under certain conditions which were present on early Earth, you can actually make organic molecules from inorganic molecules, which does not happen very often these days around us in ponds or water bodies. So that was the interesting and innovative idea. So please come to your NCRT. There is one diagram of Urey Miller's experimental setup. Let me see if I can pull that out. Yes. Can everyone see this diagram here? Is it visible, everyone?
Yes.
Or shall I make it a little bigger? Visible?
Yes.
Okay. So this was their experimental setup. Now what they did is to simulate volcanic eruptions and thunderstorms. Basically, thunderstorms or volcanic eruptions, they provide lots and lots of energy into the atmosphere in the form of heat and in the form of, like, you know, electricity. Do you understand? And the heat and the electricity combined, according to the theory, provided the much needed energy for chemical reactions to happen. In order to evolve, the elements have to constantly keep bombarding against each other, make and break bond. And this was happening over millions of years. Now, one risk was you will not get anything in this experiment, but still they thought, okay, let's do it. So they used electrodes to simulate the volcanic eruptions and thunderstorms part. And these electrodes used to discharge sparks at constant intervals. You will just discharge spark from these electrodes, and that spark, and this whole setup will be in a airtight glass vessel. And in this glass vessel, there will be gases, the exact same gases which were the part of the reducing atmosphere. Okay, so you will just pump gases, only these gases. So it's methane, ammonia, water, and hydrogen, right? These things. So second things, first, high temperature and volcanic eruptions were taken care of. Okay, and then reducing atmosphere was also created. Now they witnessed that these gases, when the spark discharge of the heat was provided, they started making reactions, they started colliding among themselves. And to cool down these gases, because over time the temperature was very high, but certain pockets were also cooling down. You know why they were cooling down? Why they were cooling down? Because the earth was solidifying, right? It was molten to begin with, but when it was solidifying, it was cooling down. Any hot object cannot stay hot forever, not even the sun. Do you know that?
Yes, sir.
That's a normal thing in science, right? You take a hot object, you take boiling water, put it in a glass and leave it. What happens?
It gets cold
Even
over
if you,
time.
yes, even if you keep it in the vacuum, over time it will start radiating heat. Heat is an electromagnetic radiation. It does not need, atmosphere to travel. That's how sun's heat reaches atmosphere. There is nothing, right? Vacuum. So over time, it will keep radiating energy in the surrounding space and will become cool. So earth was also cooling down as it was solidifying. So to mimic that, they use this water in and water out thing. So water is not interacting with these gases, okay, directly. It is just playing the part where it is cooling down, taking the heat from this pipe. So you see that this one is a constant pipe, right? This part is just on the top of it. Imagine it like anyone have seen a sphygmomanometer used to measure blood pressure? They put a band
Yes, sir.
around the arm and they
Yes.
measure the blood pressure. So nothing goes inside the body, the thing is just on the outside. So this thing, the water in and water out, is also just on the outside of the pipe. It's a closed, sealed chamber, okay? So when water is coming in and making this part cool, and then water is going out, these gases will also cool down, and lot of water vapors, this H2O, which is present in gaseous form, which is heated up, will also cool down, and water in the form of droplets will start accumulating at the base here. So this is the water which is accumulating after cooling down. This is not this water. So don't think they are putting water in the system and this is the water which is getting accumulated here, because that way you can say that the whole system was contaminated. Something just came with the water. Do you understand why I'm explaining this?
Yes, sir.
Yeah. And this, then again, when it will accumulate here, then again there will be a rod which will be heated again to make the water boil, because it's a cycle happening on earth. The water is cooling down, then again volcanic eruptions are happening, heat is coming in, and these small potholes of water which are not yet connected oceans, they are again getting heated up, evaporating. So from here the water will again evaporate, go up, but it's staying in the earth system only. So this is also a closed system. Then again it is coming down, coming in contact with spark discharge energy, again they are colliding, and this cycle is just happening like 100,000 millions time. And on prehistoric earth it was happening for millions of years. In their experimental setup they did it for months and years, and they figured out that this water over time, you just there's this tap, so you just open it, it's a vacuum tap, you open it and collect some water as sample and look what is present in the water now. And over time they found organic compounds. When they started, they only has put all the inorganic compounds, methane, ammonia, water and hydrogen, the early earth conditions, and they started finding organic compounds like sugars. And in similar experiments done by other scientists, there were amino acids found. Now, amino acids are very complex ones, right? It's not just methane or ammonia. And you know that amino acids needs amino group, right? Which comes from ammonia.
Yes, sir.
Yes. Sugars also need carbon, which can come from methane and hydrogen, right? It needs oxygen, which can come from water. It needs nitrogen, which can come from this N. Okay. So sugars, amino acids, and they started finding in this solution. Now, what does this prove? Is this a proof enough to say that yes, actually chemical evolution can happen if these conditions are met. So if it can happen in a vessel, in an airtight vessel within months and years, imagine for millions of years. If you allow this to happen, you can also get complex amino acids can go on to form complex proteins, and sugars can go on to form complex carbohydrates. Yes or no?
Yes, sir.
And also lipids. And we know that lipids can form plasma membrane, the most the vital thing about a cell that distinguishes the inner environment of the cell different from the outer environment. Do you understand this whole experiment that Miller did?
Yes, sir.
Yeah, so it makes sense. Now, do you think that it's proof enough, or your skill, or you think that no, it's not a proof enough because I still have X, Y, and Z doubts? Write this, mark these things. Is everyone convinced? So you have to be, if you are not convinced, do not let me move forward. Okay, but if you're not convinced, you have to tell me why are you not convinced. It's not like I don't feel like being convinced. So science does not work on feelings. Some things that you think cannot happen actually happens in science, and there are proofs for it. So this experiment where they figured out organic compounds in the water where there was no organic compound introduced and everything was just inorganic, they still figured out sugars, amino acids, and lipids. This is a proof enough that organic compounds can be formed as a result of an interaction between inorganic compounds. Yes or no?
Yes, sir.
Yeah. So let's write down certain things for this experiment. Miller created the early earth conditions. Miller created the early earth conditions in a laboratory scale. On a laboratory scale, where on a laboratory scale where he used electric discharge through electrodes. He used electric discharge. So do you agree that electric discharge from the electrodes mimic the same thing that happens by through thunderstorms, and it also creates heat energy that might be coming from volcanic eruptions?
Yes, sir.
Yes, because the nature of electricity is same, whether it's coming from your 5-volt battery or your car battery or your electric sockets or from a thunderstorm, from a thunderbolt, or from anywhere, electricity, or from solar panels. Electricity is electricity. The rules of electricity is, what is electric current? Physics 101 basics. I think electricity is your first chapter, isn't it? Yes. Flow of charges. So when charges are made to flow in one direction in a closed circuit, it creates electric current in that circuit, right? Yes. So what did you write?
He used
He
electric discharge.
Yeah, he used electric discharge from electrodes in a closed flask, in a closed airtight flask, so that no contamination can come in, right? Because now in the atmosphere we have lots and lots of molecules and things, viruses, bacteria, bacterial spores, fungal spores. So if anything can get into this experiment, it will just broken down and we will find these organic compounds due to that. So that's why this is a vacuum airtight, and this experiment has been replicated and repeated multiple times. Even today, if you do that, you will find the same result. So in science, apart from just doing the experiment, what also matters is that science is reproducible. That's the beauty of science. It's not magic that only few can do and others cannot. Anyone can do science. all the experiments that were ever done in science are all reproducible. If something is not reproducible, it's not considered science. Okay. That's why the concept of multiple replicates come in. You know, anything you have to prove it at least 3 times, n equal to 3. You have studied this? You all must be making science project files and copies, right? Laboratory notebooks in school.
Yeah, sir. You should get like 3 values for the
Yeah.
answer
So
to
have
be.
you know that n equal to 3 if you're doing titration in chemistry? Because I remember doing this in school. Anything that you do, you need to get
Yes.
even the physics experiment. If you're doing something, if you're trying to find the value of small g, which is the acceleration due to gravity, you need to do the experiment 3 times and write the value every time, right?
Yes, sir.
Yes.
Yeah. That is because that's the motive of science. Something in the first go you can just misinterpret, right? Maybe you just did not take into consideration a factor at all, the factor of something, and because of that you got a result and you are thinking that it is science. You applied logic, but when you do it again, the chances, if you do it 3 times, is becomes less and less that you have done a mistake. And every time if you get the same result, if every time you get different result, then also it's not considered science, right? That maybe there is something you are doing wrong, or this is not the right way to do this experiment, because the results are coming different every time. So you need to get almost like some similar results every time, where you can make a common, then you make this, plot the data on a graph and make the graph. Do you do that? So on x-axis, let's say there is something, on y-axis it's time. So at time 1 second you did the experiment and you find that the value was this, then on 2 seconds you did something and the value should be this, right? So there should be a linear trajectory. So there should be at least 3 dots to connect this linear trajectory. You must have done all of this kind of work, right, in your project files. That's the motive. They are teaching you science, essence of real science. Yeah. Okay. Write down next point. In a closed, not the next point, continue in this. In a closed flask, this is what I said the last, right? In a closed flask containing components of the earth, containing components of prehistoric reducing earth atmosphere, and in the brackets you can write ammonia, methane, water vapors, and hydrogen gas. Okay. And you know what was the temperature? So even when we talk about this high temperature, what temperature we are talking about? At what temperature do you get water vapors? At what temperature do we get water vapors?
100 degrees Celsius.
100 degrees Celsius, right? So it's obvious that the temperature must be above the boiling point of water, which is 100 degrees Celsius. That's where you can get gaseous water in the form of water vapors. Now, what could be the highest, like what happens if we go beyond 100 degrees? If I start giving a water more than 100 degrees, what happens? The steam gets more and more heated. You know, steam can be heated up to very, very high temperatures. Steam can contain that energy. So this temperature that we're talking about is around 800 to 900 degrees Celsius. Way too much than the boiling point of water. That's why this cooling down process, to fasten it up, you need water in and water out. On early Earth, it was not happening very fast. It was happening on a very, very slow scale, on a time scale of thousands and millions of years. Okay. So write down next point. Miller found that there was formation. Miller found that there was formation of organic molecules like amino acids, in the water, in the cooled water present in the flask, or liquid water, in the liquid water present in the flask. And then other people did the same experiments, and they also found presence of sugars and lipids, etc. Okay.
Okay, sir.
Yeah. And very interestingly, if I tell you, sometimes when meteorites fall on Earth, you know that meteorites do fall on Earth. Sometimes they reach the Earth's surface. And then these stones are procured by scientists and they are studied. There are meteorites that have been collected from Earth and space that contains traces of organic molecules in it. What does this tell you?
That you cannot disprove the what is that?
You cannot disprove what?
Panspermia.
Exactly. And it also tells that this is actually, yes, it can be used as a proof for the theory of panspermia, and it also can tell you that similar kind of reactions are very well possible to happen in other pockets of the universe, right?
Yeah,
But
sir.
this also does not prove that life must have evolved, because we just have proved chemical evolution. But will chemical evolution always be followed by biological evolution? That's yet not proved. And just by getting organic molecules in meteorites, you cannot say that just because this meteorite has some organic molecules containing carbon, hydrogen, nitrogen, and oxygen, then life must be present from where this meteorite came. Is it clear, Fatima?
Yeah.
Yeah. So it just says that chemical evolution is possible. The first part of the theory that was put out by Oprin and Helden. So write down.
Sir, so this is
Yeah.
also not true, right? That
What is?
in chemical evolution, biological evolution came.
That is what I'm going to prove in the next section. We are going step by step. Okay. So till now, I have just proved. So you should not, just because I told you one experiment that we figured out amino acids, you cannot say that, yes, because we have amino acids, because we have lipids, because we have sugars, now the cell will be formed. We have to prove it some other way, right? Because
Yes.
what we have to prove, what is yet left to be proven. The next part is even trickier. What is left to be proven is, okay, you have these molecules, but what now? These molecules are just present in a water soup. How can they become a cell? So the answer lies, they need to assemble together, right? Lipids needs to assemble together to make a plasma membrane. Now who's assembling the lipids? And then people came, okay, remember, so if science gets stuck here and cannot provide further proofs, then also the people who are supporting special creation can come in and say that, see, this is where you need a creator. Someone has to assemble these lipids together to make plasma membrane. Yes or no? And it's a very valid argument, actually. You
Yeah.
agree with me? Yes.
Yes.
Yeah,
So science, yes, Matthew?
yeah, it is a valid argument.
Exactly. So science, to disprove that, science has to prove that, see, no one is required to, organize these lipids. Lipids or amino acids have the property of self-assembly. They can assemble on their own under right conditions. And again, you have to prove it by experiments. Yes or no?
Yes.
If you can prove in a beaker or in a flask, like what they did, that if you have these lipid molecules and if you have these kind of organic molecules, they can self-assemble to give rise to structures, then only you can move one step forward to say that from chemical evolution, biological evolution kicked up. Yes, do you agree with me? Yes or no?
Yes, sir.
Yes, sir.
Because if that is science, again, that can also be reproducible. That should be reproducible. That no magic or no vital force or a special creation is required. It can happen on its own. So you better prove it. Otherwise, you don't. This is where science gets stuck and believe in special creation or panspermia. So let's see what happened next. So is the story till here clear? Everyone on the
Yes,
same
sir.
page? Everyone is believing, right, in science? Okay. So write down the next point, that with this limited evidence, we still call it limited evidence, okay, though it was a groundbreaking research, but yeah, groundbreaking research can also provide evidence which in a broader perspective is limited. So write down, with this limited evidence, the chemical evolution part of the theory put forth by Oparin and Haldane, the chemical evolution part of the theory put forth by Oparin and Haldane was more or less proven and accepted. So it also have to be accepted by the scientific community. Okay. Whenever someone does the scientific community is not like, yay, great. They're like, what, how, where, why? Show the data. So science is critical. The community is taught to be critical. So whenever someone like when in a PhD or in a research field, when we write something, an experiment, a thesis, we have to defend it. So there's something called defending the thesis, defending your research, where you have to sit in an open seminar with hundreds of people who can just walk in and sit there ready to rip apart your experiment, find flaws in it, ask you questions, put you in a difficult position, and you have to defend. Actually, it's a defense, okay? And the arguments can be heated, the arguments can be directed, but they're not personal. They're all based on science, okay? That they're not on you, your inability, or you're a fool, not like that. It's like the science has this flaw. Okay, your experiment doesn't lacks this thing. So this is how, you know, theses are also submitted. So everything has to go through a scrutiny. The same happened with this experiment. So it was more or less accepted, and they realized, okay, let other peoples do it. So everyone around the globe started doing this because it was a very hot topic, that, oh, Miller actually created organic molecules from inorganic molecules. Some people said very good. Some people said, he's just making a fool out of everyone. Let me try and do it. Then they did it and they followed the same protocol, find out the same results. They started accepting. So more or less, this part was accepted. But still, we have no idea till this time that how does this molecule become a cell? Because what is a cell? It is a encapsulated space. Yes or no?
Yes, sir.
Yeah. So let's move forward from here. The theory of abiogenesis, which says that life emerged from inorganic substances. So what was yet left to be proven was that chemical evolution, write down, chemical evolution led to biological evolution. Biological evolution. Okay. And it is believed that the first non-cellular. So the question came up, do we always have to make a cell to be living? Can only a cell be living? Am I audible and visible, people?
Yes.
sir.
Yeah, I'm so sorry. I lost internet for some time. Also electricity at my place. Okay, I have to reshare. Okay. Sorry for the inconvenience, people. Okay, let's. What was I saying?
Can only a cell
Yeah.
be living?
Yes, right. So tell me, what do you think? What do we know about living systems? What are the first basic criteria or characteristics that a living system should meet if it's a very simple system? The first criteria it should meet logically, that it should be able to maintain its environment separate from the environment, like its own environment separate from the surroundings. Yes or no? So it should be encapsulated. Do you agree with that?
Yeah, sir. Yeah.
Second thing, it should be able to self-replicate. It should make its copies. Otherwise, if it is just one cell that managed to evolve and then it cannot replicate, it's gone, right?
Yes.
Yes, sir.
Agree? So the ability of self-replication and the ability of self-assembly, these two things have to be proven if you want to join a link between chemical evolution and biological evolution. Yes or no?
Yes, sir.
So NCERT does not do a good job of linking this thing, but just to, because if I do not make you convinced on this step, then the point of me being your biology teacher and teaching you evolution is failed. So that's where NCERT does a very bad job. So, and also when probably it was being written, there were not many evidences that could be explained to students very simply. But now we know about a lot of self-assembling inorganic systems that assemble just like organic systems. Do you know about lysosomes in a plant cell? Lysosomes. No one knows about lysosomes in a plant cell. Everyone forgot class 11th. That's not how you are going to do good in entrance then. People, lysosomes. What are lysosomes? Or vacuole. Forget about lysosome. Vacuole.
Yes, sir.
Central big vacuole in plant cell. Plant cell has a big central vacuole, right? Animal cell have multiple small vacuoles. What is a vacuole? Any idea what is a vacuole?
It's like kind of like a storage area for the plants.
Yes.
Where...
What it is made up of? Why it is a vacuole? Why it's a bag inside a cell, right? Cell itself is a space. So it is a space inside a space. What it is made up of? Cell is made up of double lipid bilayer membrane. Okay, plasma membrane. Understood. What is vacuole made up of? Logically, what it can be made up of?
Nothing.
Nothing? It is made up of nothing. Then how it is a space?
Empty space, no?
It can be?
It's empty space, right?
But how is it empty? Can you create an empty space in a glass full of water?
No.
Then you think cell is vacuum? Cell has a vacuum inside? Cell has cytoplasm. It's a fluid filled space, right? In that fluid filled space, you have to make a empty space. How will you do that?
You have to make an enclosure to do that.
Yes. And how can we make enclosures in biological systems? Simple, through making a membrane. Mitochondria is an enclosure. Endoplasmic reticulum is an enclosure. Nucleus is an enclosure within an enclosure. A bubble. I asked Fatima this question, can you create an empty space in a glass full of water? She said, no, you can. Just create a bubble in it. Just blow a bubble. So a bubble, do you see that bubble is an empty space?
Yeah.
No, it's not an empty space. It's air inside water.
Sorry.
Yeah. So I'm just confusing you. If your fundamentals of science are not strong, then you have to work on it. Be a scientist first to challenge any theory. Okay. The other way to create a space, not empty, but a space, because vacuoles are not empty. Vacuoles contain some things. Most of the time they contain waste substances. which are excreted by the cell itself. And vacuole is made up of a membrane. And how are membranes made in biology?
Lipids and proteins.
Perfect. Lipids are the primary players. Proteins comes to enhance some functions, but in a, do you remember this word tonoplast? Studying in class 11th. Tonoplast. It is, we name a lipid membrane tonoplast, which is lipid monolayer. And when it is bilayer, we call it plasma membrane. Okay. So the membrane of vacuole is called tonoplast. Okay.
Okay, sir.
Clear? Please go back. Whenever is your entrance, before that, go back to your 11th NCERT. These things are fundamentals. You cannot forget this. This proves the presence of tonoplast in nature proves that to make an enclosure or encapsulate something, it's not always mandatory that lipid have to form bilayer. If lipids can manage to form monolayers, then also they can make enclosures. Yes or no? And the presence of tonoplast in a highly evolved setup like plant system tells that monolayers of lipid are also membranes. Yes or no?
Yes, sir.
But then what will they contain inside? Remember what a lipid structure is like? It has a head and it has a tail. This head is hydrophilic. I'm teaching you all of this because this is important. This is where NCERT takes a shortcut. It tells you in one sentence, okay, chemical evolution happened and biological evolution happened. Now let me tell you how organisms evolved. But the $1,000,000 question is, if you cannot convince someone that actually chemical evolution led to biological evolution, then it's all waste, right? Whatever you are going to tell next is just a story for that person. So the lipid has a hydrophilic head and a hydrophobic tail. What does this mean? Which means the head part can interact with water very well. Hydrophilic, they love water. Okay. But the tail part cannot interact with water. It's hydrophobic. That's why in a lipid bilayer, you always see, thanks. That's why in a lipid bilayer, you always see lipid membranes arranged like this, right? Yes or no? So this head part is towards outside and also towards inside, because inside is cytoplasm and outside is water or water-based environment. Is it clear, everyone?
Yes, sir.
So on both, and what is cytoplasm? Cytoplasm is 95% water again. So on both sides, a cell has to interact with water. Sorry, a cell membrane has to interact with water. That's where lipid bilayer becomes mandatory or necessary. But in a tonoplast, in the inside, tonoplast is a membrane like this, just like this, a lipid monolayer. So you can easily now logically understand what should be inside. Anything but not water, right? Yes or no?
Yes, sir.
And this will just continue to form a vacuole. Do you understand? So if you make a vacuole like this, all the heads will be here and all the tails will be inside. Make sense?
Yes.
sir.
And inside will be something which is not water based, trapped. Waste, enzymes, proteins, things, anything, any molecule which you hold inside, RNA, DNA, you can pack anything inside which is not water based. And this is how you can make the most simplest type of capsule in biology. Yes or no?
Yeah.
Yes. So if life has to evolve from non-living substances, you cannot in just one step make a cell. That scientists understood just by logical thinking and reasoning. Now all they wanted to do is to prove it experimentally. And you know, it's very, very interesting that we all are actually doing this same thing every day in our life. How many of you use soap? All of us use soap and detergents. Yes or no?
Yes, of course.
Yes.
In chemistry, have you heard of this one term called missile? What is soap made up of? Either it is made up of animal fat, like when it was, now they are synthetic fats, but animal fat or plant-based fat, right? Or synthetic. Basically, it is lipid. Do you understand? Soap has lipid molecules, different types of synthetic lipid molecules with aroma, esters to give you rose flavor. All these are just commercial things. Basically, as a function, these are lipid molecules. Do you understand, everyone?
Yes, sir.
Yes. Now, you know, how do soaps do their cleaning actions? How can soap clean your water? Oh, sorry, clean your clothes or your hand. So your hand has some dirt, and now you are using soap and your hand is clean. You know what happens? So it happens through micelle, and micelle is very much like a tonoplast. So what happens is these lipid molecules, the hydrophobic part. So, and soap always works with water, right? There is no soap which works without water. They are called sanitizers. They are not cleaning agents. Sanitizers, hand sanitizers don't clean your hand. They kill the germs present on the surface of your hand. That's it. But after killing, the dead bodies are there only. There's a difference between washing and sanitizing. Do you know the difference? Now do you understand, everyone?
Yes, sir. I understand it now.
Yes, sir.
Yes. So hand sanitizers, when you apply, your hand is not being cleaned. Your hand is being sanitized. If there is a pathogen that can cause disease, now it's dead, because sanitizers contain ethanol, alcohol. Alcohol burst or dehydrate the microorganisms, because microorganisms can only survive in water, not in alcohol. Okay. That's why all your medicines, the tablets, not the capsules, the tablets are alcohol based. All the tablets are actually, if they're not steroidal tablets, I don't know about steroidal ones, but mostly the tablets. So it's a powder, right? How is it stick together? It needs to be put in a solvent and molded in a, so they are all alcohol based. Now coming back, so micelles are these monolayers. So what they do, they have a hydrophobic tail. So this hydrophobic tail of every lipid interacts with the dirt, because dirt particles are not water based. Dirt, any sort of dirt on your face, most of all the dirts are oil based. So they interact with the dirt and happen to make a 3D. Now, can you imagine, all the tails are interacting with the dirt, right? And all the heads are towards the outside. Now imagine a 3-dimensional ball made up of pinheads. You know a pinhead? A pin with a head?
It's like virus.
Yes, you are imagining the structure, right? You are able to imagine the structure.
Yes.
It's a 3D structure where all the lipid heads are towards the periphery, and they're so tightly packed that they cannot allow water to go in, because heads can interact with water but tails cannot. So all the tails are packed inside and they are trapping dirt in between. So these are the lipid molecules of the soap, and this is the dirt which is trapped in between. So that's how all the dirt can be removed from the surface and trapped in the form of lipids. Okay, it's a monolayer membrane, just like a tonoplast. And then when you wash it with water, all the heads interact with water. They become soluble in water. So basically the micelle is soluble in water, but the dirt remains trapped in that. So when you agitate the clothes after washing, when you put it in water and agitate it, you are actually getting rid of all the dirt. Same is with hand wash or soap. You apply it on the surface, all the dirt gets trapped, then you wash it with water, everything is washed away. If you just wash it with normal water, the dirt might not go away, because it's also stick to the surface. So these things take it out from the surface and trap it within the micelles. So this kind of arrangement of lipids is called a micelle formation in soap. Do you understand, everyone?
Yes.
So
Yes.
I'm teaching you some chemistry here basically, but it was, I think, important if your basics were not clear. So now is it clear? So what's basically happening here in soaps? Aren't we witnessing a self-assembly of lipids?
Yeah.
Yeah, on a very low scale, like it's just doing one function of trapping dirt, but aren't we proving basically that lipids can self-assemble?
Yes.
Yes. People, Say yes or no.
Yes,
Yes,
sir. Yeah.
Okay, very well. Similar experiments, which are not, many experiments have been done in space, in International Space Station. People did some experiments where in vacuum, they also saw that certain inorganic and organic compounds behave differently, and they, tend to attach together. They tend to come together and assemble. Another example of assembly of inorganic substances is making of crystals. Have you seen crystals ever? Crystals, who make crystals? Crystals assemble on their own in nature, right?
Yeah.
Yes, sir.
And
Crystallization.
yes, and every crystal, or snowflakes, you must be knowing that snowflakes have a particular shape. The microstructure of a snowflake is same, you know. So this assembly of organic compounds to make bigger structures is now very well proved. And so first thing, that a membrane can be made from lipids. Yes, it can be. If you give millions of years in the right condition and lots of lipid present somewhere, they can self-assemble to make a membrane. But the first kind of membrane will not be bilayer all of a sudden to give rise to a cell. It will be a monolayer to give rise to something like a vacuole kind of structure. And in that vacuole, then you cannot fill cytoplasm. You can only put some things that are not water based, like RNA, protein, or something like that. But does this will this behave like a living system? That is the next question that we have to prove. So till here, do you understand that yes, self assembly can happen, but it will give rise to a very simple monolayer, lipid monolayer system containing RNA or something inside. Is it clear till here?
Yes, sir.
Now, next that scientists have to go forward to prove this is that, can a system with just an enclosure and some basic macromolecules or some basic organic molecules inside, can it start behaving like living system? Can it replicate? So self-assembly part is proven. Now, self-replication part has to be proven, because bacteria self-replicates. But bacteria, believe me, is very advanced an organism when we are talking it with respect to a tonoplast or these kind of early structures. You understand? So everyone believes that in those water pots, in the Urey-Miller experiment, one day you will find bacteria-like cells. No, to find a bacteria-like cells, you need millions of years, because bacteria are very complex. But you can find these simple things over thousands of years, because we can do it in synthetic chemistry. But now the question is this kind of a system living? Can it self-replicate? So the answer is yes. There are many infectious agents in our surrounding, and one of that infectious agent, where the coat instead of lipid is made up of protein and it only has RNA or a DNA inside it. It behaves like living under certain special conditions. It can replicate, and you know it. It is creating a global pandemic nowadays. What is it?
COVID.
Sorry? Sorry, Matthew, what did you say? You're not audible if you're speaking, I think.
Viruses.
Exactly, viruses. But earliest life could not have been like viruses. Can you tell me why? There's a problem. If you say that earliest life was like a virus, just a protein coat with a, earliest life has to have a lipid coat, not a protein coat. Can you tell me why? The answer is in the virus. How does virus replicates? Can a virus replicate without a host cell?
No.
No, right? So viruses need host cells. Now, when we are talking about the first cells who were living themselves, there is no host cell for, if viruses evolved the first, then it does not make sense. There is no host for those viruses. Is it clear? Do you understand?
Yes.
So it has to be lipid based. And so the answer again is yes, because there are many simple substances which are just lipid based and they can replicate. Okay, now that mechanism is a little convoluted, but it has been found that's very simple systems containing membrane, lipid membrane, can replicate. Okay, even which are even simpler than bacteria, even primitive than bacteria, it has been found. Everyone understands?
Yes, sir.
Okay, so write down the first form of life. The first form of life. Now, let me also write it here. The first form of life should have been self-replicating metabolic capsules which are still non-cellular. So the concept of cell is also advanced. So before that, we need to, it was a very big jump. That's where people stopped believing in abiogenesis, because at one point it was easier to prove chemical evolution. It was easier to prove chemical evolution. It was easier to prove that one cell can divide to form many. But how can from chemicals a cell can be made was a very big leap. That was not at all logical in science. So there has to have something intermediate, and they found it that even non-cellular aggregates, just as simple as a membrane and certain organic molecules inside, also do exist, and they are able to self-replicate. Okay, and they are not viruses, they are, because viruses need a host. So this bridged the gap. So now the jump is not very high. It bridged the gap, and this theory was again back into acceptance in science, and people started finding more and more proofs, doing experiments like synthetic chemistry. And the most simplest kind of example is a missile formation, where lipids can actually self-assemble. Okay, and write down next point.
So how do the viruses form if they need
Yeah,
a,
viruses. Sorry, if they complete
if
your
they
question.
if viruses need a host to survive, then how were
Yeah,
they formed?
yeah. So it is. So they can only be formed after the host, right?
Yeah, exactly.
Viruses
So
cannot form before host.
how did they come?
Yes. So it is now proven. It's not just viruses. Viruses are not very special. We consider viruses special because we have not studied about them much. Even proteins, naked proteins can cause infection. Just proteins can cause infections, and these are called prions. Go and read about. Science is very much advanced now. We have figured out multiple answers which very few people know. If you just have to go into higher studies to need this thing. But there are prions, there are virions, which are even primitive, and viroids and viruses. Viruses are not just the only thing. So if in nature somehow any nucleic acid can get coated by a protein, that can become a virus. Okay, and because nucleic acids have this property that they can be read by ribosomes. If it is a RNA, it can be read by ribosomes and can be translated into protein. You understand?
Yes, sir.
So that's where viruses evolve. So exchange of nucleic acids between one and the other organism constantly over millions of years. So earlier people used to believe that viruses are very, very primitive. They came before, but just like you said, it does not make sense. If viruses came before the host, where were they replicating? Yeah,
Exactly.
So they came after. So these earliest form of self-replicating structures were not viruses. They were something made up of lipid only, because lipids show this property of self-replication, not proteins. You understand? and virus
Yeah.
coat is made up of protein, not of lipids. Is it clear?
Okay, sir.
Yeah.
Yes, sir.
So that's why all the life on Earth, which is cellular, okay, has a plasma membrane. That's important. You may or may not have cell wall. You may or may not have vacuole, but you have a plasma membrane. Yes, agree with me?
Yeah.
That's where this theory again can be proven, that if it was not for self-replicating structures, then why do all cells have plasma membrane? Regardless whether it's a bacteria, fungi, protozoa, plant, or animal, all have plasma membrane made up of lipids. Okay.
Okay, sir.
So you can, in your free time, if you want to learn more about the intricacies of biological systems, go read about these prions, virions, viroids, viruses. Viruses you must be knowing because it's a hot topic from last 3 years. Anyways, so the first form of life should have been self-replicating metabolic capsules, which are still non-cellular, right? And then write
Yes, sir.
down what they would have been. So these would have been giant molecules, or not, don't use the word giant. These would have been macromolecules. These would have been macromolecules, because this can be experimentally proven. Macromolecules like proteins, nucleic acid, sugar, etc. encapsulated by a membrane, by a lipid membrane. Now it was monolayer. If you can form monolayer from lipids, then I think I don't have to go and explain that from a monolayer a bilayer can also form, right? And actually experimentally there are many labs working around the world. To make a bilayer out of a monolayer, you just have to break the monolayer and fix it. Like if you break it, in fact, if you break the monolayer, they have this tendency of folding upon itself and making a bilayer. So that's not difficult to understand, right? Convinced? Not convinced?
Yeah, kind of convinced. Yeah.
So basically what I'm talking about is that a monolayer is kept like a monolayer because there is a surface tension. These lipid molecules are like this. All the heads are here and all the tails are towards inside, right? Something has to be inside which is being trapped. Okay, non-water based. Now if I just break it from here, this monolayer will just become straight, and then it has a tendency to just fold over itself. So these are the head and these are the tails. Does it become bilayer now?
Yes, sir.
You understand or not?
Understood, sir.
Very good. So, but write down the first cellular form of life. So we reached till here. Is the story making sense to you? Are you enjoying it? Oh, okay. I think we are way much ahead of the time. Okay, people, we have to stop here. So we'll continue from cellular form of life in the next class. The topic was interesting, so I lost the track of time. Okay, so I'll see you all in the next class. Please come with lots of doubts, and we'd love to have a discussion. Okay.
Okay, sir. Okay, sir.
See, if you challenge me, I'll challenge myself to give you better proofs and things. If you don't challenge me, I'll just regurgitate NCERT, and you may not be convinced about evolution at the end. Okay.